The prebiotic atmosphere of the earth was of a reducing nature. It was transformed into oxidizing atmosphere of the present day due to the emergence of cyanobacteria. Perishable vegetables are sprayed with a solution of cytokinin. Genetically modified organisms, the DNA of the organism is altered. The cutting organ in the mouthparts of cockroach is mandible. Hello everyone, myself B. Shobha Rani from the Department of Biology, Vidhyaashram Pre-University College, the Temple of Excellence. So in my today's session, I'll be doing the analysis part of Karnataka CET 2012 Biology Analysis. Biology paper analysis. Karnataka CET 2012 Biology paper analysis. Moving on to the question number 1. Which of the following is not a character of cancerous tissues in our body? So, which among the four, which is not the character of the cancerous tissues in our body? Option 1, contact inhibition. Option 2, neoplasia. Option 3, metastasis. Option D, inability for differentiation. What is contact inhibition? Ceasing of cell division. Neoplasia is uncontrolled growth of cells. Then metastasis spreads, the cancerous cells will not remain in the region where they have originated. They keep spreading to the other areas, and that process is called as metastasis. And inability for differentiation. Differentiation means what? Process by which cells, tissues and organs acquire specialized features, especially in the embryonic development, but the inability for differentiation. So which of the following is not a character of cancerous tissues in our body? This is the character of a cancerous tissue. This is also a characteristic feature of a cancerous tissue. This is also character. But which is not a character is contact inhibition. What is contact inhibition? As you know, contact inhibition is the ceasing of cell division and multiplication when it comes in contact with another cell of the tissue. So when new cells keep on adding, the neighboring cells will prevent the multiplication of cells. But that quality will not be found in the cancerous cells. And actually you can call it as a genetic programming that occurs in normal cells, which is not found in the cancerous cells. And the cancerous cells, so in cancerous cells, this feature is mutated, resulting in an uncontrolled growth. So it will not inhibit the growth or the multiplication of cells, whereas the normal cells do that process. And this makes them what? Immortal. So, they keep on multiplying and there is more growth of cells will remain forever. So, like they keep on spreading. So, it means they can't stop by contact inhibition. So, which of the following is not a character of cancerous tissues in our body? It is option 1 called contact inhibition. So, option A is the correct answer. So, which of the following is not a character of cancerous tissues in our body? It is contact inhibition. Moving on to the question number 2. Which of the following statement is not true for Nostoc? Which of the following statement is not true for Nostoc? Option A, it is prokaryotic. Option B, it is autotrophic. Option C, it is filamentous. Option D, it is macroscopic. As we know that Nostoc is a microscopic algae. It is not a macroscopic algae, very microscopic algae, and it is filamentous and it is cylindrical. They are nothing but cylindrical structures. They are prokaryotic cells and they are autotrophic in nature. So all the characters are there here. But which among the four is not true for Nostoc? As I said, it is prokaryotic, correct. It is one of the feature of Nostoc. It is autotrophic, this is also correct. It is filamentous, so this is also a feature of Nostoc, but which is not a feature, macroscopic. It is wrong. It is microscopic. So which is not true? It is option D. Option D, that is, which of the following statement is not true for Nostoc? That it is macroscopic. Moving on to question number 3. The system of classification of plants proposed by these two botanists is claimed to be a natural system. I repeat, the system of classification of plants proposed by these two botanists is claimed to be a natural system. Option A, Engler and Prandtl. Option B, Bentham and Hooker. And option D, Aristotle and Theophrastus. And option D, Darwin and Wallace. As we know, Engler and Prandtl, they are responsible for phylogenetic system of classification. Phylogenetic system of classification. So the system right now what we are following for the classification of plants is based on the two very important scientists by name Engler and Prandtl. Bentham and Hooker are the scientists who classified, that is classification of plants proposed by these two botanists claimed to be a natural system. These were the scientists who brought about the classification of plants called as natural system, right? Next, Aristotle, I think all of us know he is called father of biology, and Theophrastus is called father of botany. Then Darwin and Wallace, they were responsible for what? They were the ones who proposed what we call it as origin of species. So the system of classification of plants proposed by these two botanists is claimed to be a natural system, that is Bentham and Hooker, that is option B. George Bentham and Joseph Dalton Hooker are the British botanists. They were the two British botanists, and classification system is based on, as you know, natural system, artificial system of natural system of classification, artificial system of classification, phylogenetic system of classification is there. But this natural system of classification, artificial system of classification was proposed by Aristotle. So here, the classification system is based on natural system, and this system includes what? The characters of plants in natural conditions according to the relative affinities. So the system of classification of plants proposed by these two botanists is claimed to be a natural system is option B, that is Bentham and Hooker. So George Bentham and Joseph Dalton Hooker. Moving on to question number 4. Match the entries in column 1 with those of column 2 and choose the correct answer. I repeat, match the entries in column 1 with those of column 2 and choose the correct answer. Column 1 includes the name of pollination, what is the name given for such type of pollination, and the column 2, the type of pollen. How is it? What are the agents which bring about that type of pollination? So cleistogamy, geitonogamy, entomophily, xenogamy, insect pollination, bud pollination, pollination between flowers in the same plant, wind pollination and cross pollination. Cleistogamy will be what? Among these four, it is bud pollination. Then geitonogamy, pollination between flowers in the same plant. Then entomophily, insect pollination. Then xenogamy is nothing but called what? Cross pollination. So A is N, B is O, then C is M, and D is Q. Now let us see which is the option which is having AN, BO, CM, DQ. Moving on to the first option, AO. So the first, this one only is wrong. Then AM, second one also is wrong. Here it is AM. Here it is AN. Here you can see AN, correct. Here you can see AQ. So this is also wrong. So obviously here you can see AN, BO, CM, DQ. So option C is correct. Option C is correct. Cleistogamy, bud pollination. Geitonogamy, pollination between flowers but in the same plant. Then entomophily, pollination through insects. Then xenogamy, cross pollination. So option C is correct. Moving on to question number 5. The host for Cercospora personata belongs to this family of angiosperms. So as we know that what are the options? I repeat, the host for Cercospora personata belongs to this family of angiosperms. Option A, Graminae. Graminae, as you know, it is also called as Poaceae, that is monocotyledonous family. And Leguminaceae, you know, we use a word called pulses. Then Malvaceae, we know hibiscus. Asclepiadaceae is also called as milkweed family. And here you can take an example of like groundnut also, right? So the host for Cercospora personata belongs to this family of angiosperms. As we know, Cercospora is a fungi. It is called fungus imperfecti. That is, it belongs to the class called Deuteromycetes. Then here they are plant pathogens. What do you mean by plant pathogens? They cause diseases in plants. And one species called Cercospora personata causes leaf spot, or it is also called as Ticca disease in groundnut family. And groundnut family is nothing but called what? Leguminaceae, which is characterized by large and brown spots on the upper surface of the leaf, followed by the separation of the cells of lower epidermis from mesophyll. So obviously the host for Cercospora personata belongs to which family? That is, the host belongs to Leguminaceae. Groundnut is nothing but belonging to Leguminaceae. So option A, Graminae, it is wrong because it is monocotyledonous. And even Malvaceae is dicot. So here Leguminaceae is correct. Malvaceae is wrong and option D is wrong. So the host for Cercospora personata belongs to Leguminaceae family. That is, option B is correct. Moving on to question number 6. The final stage in the tissue culture program before the new plants are taken out for cultivation in the fields is known as. I repeat the question. The final stage in the tissue culture program before the new plants are taken out for cultivation in the fields is known as. Option A, micropropagation. Option B, hardening. Option C, called calogenesis. And option D, embryogenesis. You know, the process of producing a new plant from taking cell from any part of a plant body, you know, it is called micropropagation. And calogenesis, hardening, that is what? The final stage in the tissue culture program before the new plants are taken out for cultivation in the field is known as hardening. Why? As we know that there are various stages in the development of the plant by using tissue culture technique. The final and the last stage is called as hardening. So there are different stages for the development of plant by using tissue culture, and the final and the last stage is called hardening, where the plants are removed from the culture medium and they are cultivated in the field. So this is the stage where the new plants are ready to be planted out in the field. And as I said, this stage is known as what? Hardening. So in this stage, the plants are removed from the culture medium and planted out in the field. So the final stage in the tissue culture program before the new plants are taken out for cultivation in the field is known as what? Hardening. Option B is correct. So here, as I said, it is known as what? Hardening. So option B is correct. Moving on to question number 7. An osmometer is filled with 0.5 m solution of NaCl in water. In which of the following solutions it must be immersed in order to make it shrink? I repeat, an osmometer is filled with 0.5 m solution of sodium chloride in water. In which of the following solutions it must be immersed in order to make it shrink? Option A, 0.5 m solution. Option B, 0.05 m solution. Option C, distilled water. Option D, 0.75 m solution. As we know that an osmometer is filled with 0.5 m solute of NaCl in water, and the osmometer should be immersed in a solution with more concentrated solution. So the osmometer should be immersed in a solution with a more concentrated solution. So this more concentrated solution means what? Hypertonic solution, and which will cause what? Exoosmosis. Exoosmosis means what? Exit of water from the osmometer. So this will result in the shrinkage of osmometer. So exoosmosis has to happen, and that is only when it is immersed in a solution with more concentrated solution. So the osmometer should be immersed in 0.75 m solution of sodium chloride, and if it is immersed in 0.75 m sodium chloride in water, this will allow the flow of water from the osmometer to the outside in the hypertonic solution. So obviously it will shrink. So which is the right option? Option A, 0.5 m is wrong. Option B, 0.05 m solution is wrong. Distilled water is wrong. Option D is correct. So option D is correct. That is, it should be immersed in 0.75 m solution. So option D is correct. Moving on to question number 8. Perishable vegetables can be maintained fresh for a longer period by spraying on them with a solution of. I think all of us know perishable vegetables are those vegetables which get spoiled very soon, right? So if you want to keep them fresh for a longer time, what is that solution which needs to be sprayed for the vegetables which are going to perish very fast? Option A, ABA, that is abscisic acid. Then option B, that is cytokinin. Then option C, ethafone. And option D, phenyl mercuric acetate. As we know that one very important hormone called cytokinin, which is a growth promoting hormone, they promote what? Cell division in plants and also slow down the process of senescence, aging. Right. So perishable vegetables are sprayed with a solution of cytokinin. Why? To keep them fresh for a longer time and increase their shelf life. What do you mean by shelf life? The longevity. So without being spoiled, they can be allowed to remain fresh for a longer time. So they also prevent what? Chlorophyll degradation. So option A, ABA is wrong. Option B, cytokinin is correct. Option C, ethafone is wrong. And option D, phenyl mercuric acetate is wrong. So option B is correct. So perishable vegetables can be maintained fresh for a longer period by spraying on them with a solution of cytokinin. Option B is correct. Moving on to question number 9. The prebiotic atmosphere of the earth. What do you mean by prebiotic atmosphere? The first atmosphere of the earth is called as prebiotic atmosphere of the earth, was of reducing nature. You know, that was the reason why life forms were not found. But now it is an oxidizing atmosphere, having more of oxygen. So it was transformed into an oxidizing atmosphere of present day due to the emergence of, I repeat, the prebiotic atmosphere of the earth was of reducing nature, having less of oxygen and more of hydrogen. It was transformed into an oxidizing atmosphere of present day due to the emergence of option A, cyanobacteria, option B, angiosperms, option C, photosynthetic bacteria, option D, eukaryotic algae. So the answer for this is cyanobacteria. As you know, cyanobacteria is also called as blue-green algae or photosynthetic prokaryotes. And these are the organisms that evolved million years back. So they were the which have evolved million years back. So they perform what? Oxygenic photosynthesis, where they used water to get hydrogen and released oxygen in this process. So they were the ones to release oxygen, and they are responsible for the present day oxidizing atmosphere. So they added oxygen to the atmosphere and paved the path of evolution of aerobic forms. So those organisms which require what? Oxygen, including aerobic bacteria. So the prebiotic atmosphere of the earth was of a reducing nature. It was transformed into oxidizing atmosphere of the present day due to the emergence of cyanobacteria. So option A, cyanobacteria, is correct. Option B, angiosperm, is wrong. Option C, photosynthetic bacteria, is wrong. Option D, eukaryotic algae, is wrong. So option A is correct. Moving on to question number 10. Match the contraceptive methods given under column 1 with their examples under column 2. Select the correct choice from those given below. Column 1 is contraceptive methods and column 2 are the examples. 1A, chemical. B, IUDs, intrauterine devices. C, barriers. D, sterilization. Here, tubectomy and vasectomy, copper T and loop, condom and cervical cap, spermicidal jelly and foam, coitus interruptus and calendar method. Chemical is what? Usually it is spermicidal jelly and foam. Right. Then IUDs is what? Copper T and loop, that is Q. Then barriers. Barriers is what? Condom and cervical cap. Then sterilization, tubectomy and vasectomy. So A is yes, B is Q, C is R, D is P. So which is the option which is having the combination of AS, BQ, CR, DP? Chemical, spermicidal jelly and foam, IUDs, intrauterine devices, copper T and loop, barriers, condom and cervical cap, then sterilization, tubectomy and vasectomy. Now we'll check the options. AS, here you can see AS, correct. Here it is also AS. Here it is AP, wrong. Here it is AS. Go for the next one. BQ. Here also BQ is there, correct. Here it is BT. So option this is wrong. This is wrong. And here BQ is there. So this is correct. Then go for the next one. CR. Here you can see CR, correct. Here CT it is there. So it is wrong. Then DP. Here you can see DP, correct. So option, so this is also wrong. Option A is correct. B is wrong, C is wrong, and D is wrong. Moving on to question number 11. One of the following movements in our body is not completely involuntary. Identify it. What do you mean by voluntary and involuntary? Voluntary functions are those which are controlled by the will. Involuntary functions are those which cannot be controlled by the will. So, one among the following movements in our body is not completely involuntary. So, it is voluntary also. Identify it. Option A, deglutition. Then, you know what is deglutition? Swallowing the food is called as deglutition. Peristalsis, that is the food when it moves inside the esophagus. Then systole of the ventricles, it has nothing to do with the heart, this one. Then dilation of pupil of the eye. Here systole of the ventricles, it is not completely involuntary, right? And even the dilation of the pupil of the eye is also involuntary. Now, one of the following movements in our body is not completely involuntary, and that is deglutition. As you know, deglutition is nothing but it is an act or process of what? Swallowing. And it involves how many stages? 3 stages. One is called the oral stage, where you take in the food, then you chew it, then it is going into the food pipe. From there it goes to the esophagus, right? From there it goes to the stomach. So the oral stage, where you take in the food, it is all, as you know, it is involuntary. So the oral stage is voluntary. the oral, when you take in the food, it is voluntary, it is not involuntary, right, in nature. So the muscles are involved in what? Mastication, chewing of the food, and the tongue helps in what? Pushing or rolling the small food of a small ball of food, which you call it as bolus, into the throat region. So it is deglutition. So option A, deglutition, is correct, and option B, peristalsis, is wrong. Option C, systole of the ventricles, is wrong, and option D, dilation of the pupil of the eye, is wrong. So option A is correct. Moving on to the question number 12. This is not a GMO, genetically modified organism. Option A, BT brinjal. Option B, golden rice. Option C, Tracy. And option D, Dolly. So as we know that in genetically modified organisms, the DNA of the organism is altered. So gene manipulation is done, and in BT brinjal, is inserted with a new gene for cry protein, and this is particularly to protect the plants from pests like the bollworm. In golden rice, golden rice is introduced with a gene which produces vitamin A, which reduces the chances of people losing their eyesight. So this is also a genetically modified organism. Tracy is the modified sheep produced by genetic engineering and able to produce the human protein alpha antitrypsin in her milk, which is used for therapeutic products. So this is also GMO, this is also GMO, this is also GMO. But Dolly is the first sheep which was made by cloning techniques, not by, it's not a genetically modified, it is by cloning technique. So option A, BT brinjal, is wrong, which is not a GMO. So BT brinjal is a GMO. Golden rice, wrong, it is also a GMO. Tracy, it is also a GMO. Dolly is not a GMO, is the right answer. So option D is correct. This is not a GMO. Dolly is not a genetically modified organism. Moving on to the question number 13. The site of Krebs cycle is option A, cytoplasm. Option B, mitochondrial matrix. Option C, intermembrane space of mitochondria. Then option D, Racker's particles. As we know, Krebs cycle is also called as what? Citric acid cycle. And here the energy is released by the breakdown of the molecules like the carbohydrates, proteins and fats. And here the energy is released in what form? It is released in the form of ATP. And the reaction of this cycle is carried out in the mitochondrial matrix. Why? Because the enzymes for this process is found in the matrix of mitochondria. So the site of Krebs cycle is what? Mitochondrial matrix or matrix of mitochondria. So option A, cytoplasm, is wrong. Option B, mitochondrial matrix, is correct. And option C, intermembrane space of mitochondria, is wrong. And option D, Racker's particles, is wrong. So option B is correct. So the site of Krebs cycle is the mitochondrial matrix. Moving on to question number 14, which is the cutting organ in the mouth parts of cockroach. Option A, labium. Option B, maxillary pulp. Option C, mandible. And option D, labrum. As we know, the mouth parts of cockroach are very unique, and they are also called as what? Trophy structures. They are also called as trophy structures, which are made up of 5 very important parts. One called labrum, mandible, maxilla, labium and hypopharynx. So as I said, labrum is called upper lip, labium is called lower lip, mandible is used for cutting the food. So it is a cutting organ, we can say. Maxilla to hold the food. Hypopharynx, the tongue of, it is otherwise called tongue. So among the 5 parts, which is the cutting organ in the mouth parts? It is mandibles. Mandibles are used for cutting the food. So here labium, option A, labium, is wrong. Option B, maxillary pulp, is wrong. Mandible, C, is correct. Labrum is wrong. So option C is correct. So the cutting organ in the mouthparts of cockroach is mandible. Moving on to question number 15. If this enzyme were to be absent in a small intestine, digestion of proteins in our body would be severely affected. I repeat the question. If this enzyme were to be absent in a small intestine, the digestion of proteins in our body would be severely affected. Option A, pancreatic amylase, which is mainly involved in the digestion of carbohydrates. Amylase means what? Next, maltase, it's an enzyme again which is very much important for the digestion of carbohydrate. Lipase, lipids, then enterokinase for protein. Very important. So, if enterokinase will be absent in a small intestine, as you know, the final process of digestion takes place in the small intestine, and particularly the first part of small intestine called what? Duodenum. So the digestion of protein in our body would be severely affected. So if enterokinase enzyme is absent in a small intestine, then definitely the protein digestion would be affected. So this enteropeptidase is secreted by the cells of duodenum, digest proteins, convert trypsinogen, inactive trypsinogen to trypsin. As you know, it acts on what? Proteins. It's an enzyme. Trypsin is an enzyme which acts on proteins, which results in the activation of other pancreatic enzymes. So it is enterokinase. The enzyme is enterokinase. So option A, pancreatic amylase is wrong. Option B, maltase is wrong. Option C, lipase is wrong. Option D, enterokinase is correct. So if this enzyme, the enzyme is enterokinase, were to be absent in a small intestine, digestion of proteins in our body would be severely affected. Moving on to question number 16. The frequency of heartbeat in our body is maintained by option A, AV node, auriculoventricular node, then SA node, sinoatrial node, node of Ranvier, and chordae tendineae. I think all of us know that sinoatrial node is called as the battery of the heart, or it is otherwise called as the pacemaker of the heart, which sets the rhythmic pace, that is the rhythmic heartbeat. And what is it? It is a specialized cardiac muscle located near the entry of upper wall of right atrium or right auricle, and almost near the entry of the vena cava, the superior vena cava, near the vena cava. And this SA node is responsible for heartbeat. As you know, it sends impulses to AV node, and AV node will allow the walls to open, right? And it is, as I said, it is responsible for heartbeat. The impulses arriving at the node accelerate or decrease the heartbeat. So the frequency of heartbeat in our body is maintained by SA node. Option A, AV node is wrong. Option B, SA node is correct. Option C, nodes of Ranvier is wrong. Option D, chordae tendineae is wrong. So option B is correct. Moving on to question number 17. Hypothalamus of the brain is not involved in this function. Option A, sleep-wake cycle. Option B, osmoregulation and thirst. Option C, temperature control. And option D, accuracy of muscular movement. As we know, hypothalamus is called master of master gland, which controls pituitary. Hence, it is called master of master gland. As you know, it is a part of brain where the pituitary gland is located. And the hormones secreted by hypothalamus, I think oxytocin and vasopressin, you might have studied that the oxytocin and vasopressin is received by the posterior pituitary, right? So here the hormones, the various hormones of hypothalamus have various functions like temperature regulation, sleep-wake control cycle, osmoregulation, homeostasis, thirst, hunger and mood. So, which is not the function of hypothalamus? Sleep-wake cycle, it is the function of hypothalamus. Osmoregulation is the function of hypothalamus. Temperature control is the function of hypothalamus. Accuracy of muscular movement is not the function of hypothalamus. So, option D is correct. Hypothalamus of the brain is not involved in accuracy of muscular movement. Moving on to question number 18. The Hardy-Weinberg principle cannot operate. Option A, if the population is very large. Option B, frequent mutations occur in the population. Option C, the population has no chance of interaction with other populations. And option D, free interbreeding occurs among all members of the population. So Hardy-Weinberg equilibrium cannot operate if there are changes in the allelic frequencies in the population. So there are various factors like capable of changing the allelic frequencies. So what are those allelic frequency, like the gene migration or gene flow, genetic drift, then mutation, genetic recombination and natural selection. These are all the various factors which are capable of changing the allelic frequency, due to which the Hardy-Weinberg principle cannot operate. So the population is very large is wrong. Frequent mutations occur in the population is correct. The population has no chance of interaction with other population is wrong. Free interbreeding occurs, option D is wrong. Free interbreeding occurs among all members of the population. So option B is correct. The Hardy-Weinberg principle cannot operate if frequent mutations occur in the population. Question number 19. The adult animal in this phylum is radially symmetrical, but its larva exhibits bilateral symmetry. Option A, Echinodermata. Option B, Coelenterata. Option C, Arthropoda. And option D, Protozoa. So the phylum where you come across, the adults are radially symmetrical. Adults are radially symmetrical and the larvae are bilaterally symmetrical. You can see this feature in a phylum called Echinodermata. The larval in the phylum, in which phylum you come across this unique feature where the adults are radially symmetrical and the larvae is bilaterally symmetrical. What do you mean by bilateral symmetry? You can cut the body of an animal into two equal halves in one plane. Radially symmetrical, you can cut the body of an animal into two equal halves in more than one plane. And larva will be bilaterally symmetrical, and when it metamorphosizes into adult, it becomes radial symmetry. So the option is Echinodermata. You can the phylum which exhibits radial symmetrical in adult and in larva it is bilateral symmetry is phylum Echinodermata. Option A is correct. Option B, Coelenterata, is wrong. Option C, Arthropoda, is wrong. Option D, Protozoa, is wrong. So option A is correct. Moving on to the 20th question. Identify the sense codon from the following. So what is sense codon? A codon which codes for a particular amino acid. Option A, UGA. Option B, AUG. And option C, UAG. And option D, UAA. UGA, then UAG, then UAA. They are called as nonsense codons, or they are also called as terminator codons, which is not going to code for any amino acid and they stop the process of protein synthesis. So hence the name, they are not going to code for any amino acid, hence they are called as nonsense codons. So among the four, UGA, UAG, UAA are called as nonsense codons or terminator codons. Terminator means what? If it comes, and AUG is always an initiator codon. The process of protein synthesis will always start with AUG. Usually it will be methionine. And so always the protein synthesis will start with AUG, which is going to code for an amino acid called methionine, whereas these three, they don't code for any amino acid. They are otherwise called as nonsense codons or terminator codons. So identify the sense codon from the following, which is going to code for a particular amino acid. So UGA, wrong, it is a nonsense codon. AUG is correct, it is a sense codon. Then UAG is a nonsense codon and UAA is a nonsense. So the option B is correct. Identify the sense codon. The sense codon is AUG. What is it? It is an initial codon which is going to code for a particular amino acid called methionine. Hope you have understood all the 20 questions what I have dealt in this Karnataka CET 2012 biology paper analysis.